Hi, everyone. I would like to thank the organizer for giving me this opportunity to present our research. I will discuss our finding concerning a transcription factor alpha-1 ACT in cerebellar development. Before I introduce alpha-1 ACT, I would like to talk about the CACNA1 gene. CACNA1 gene is located at chromosome 19p13. Initially, we only recognized that CACNA1A gene encoded the voltage-gated calcium channel subunit alpha-1A, which is involved in pre- and post-synaptic calcium signaling. The first association of CACNA1A with human disease dates back to 1996, as of described CACNA1 mutations in two neurological disorders, familial hemiplegic migraine type 1 and episodic ataxia type 2. In 1997, a third disease, the spinocerebellar ataxia type 6, SCA6, was mapped to CACNA1A locus. This is CAG repeat in C-terminal coding region, expanded in SCA6 from a normal range 418 to disease level 19 to 33. Differently from the proximal CACNA1A disorder, SCA6 is a later onset disease manifesting as an isolated cerebellar ataxia and characterized by the progressive cerebellar Purkinje cell loss. Initially, SCA6 pathogenesis was thought to stem from the dysfunction of voltage-gated calcium channel. However, experimental animal models expressing mutated alpha CACNA1A failed to consistently yield deficits in channel function. The spreading of next-generation sequencing technique linked de novo CACNA1 virus to an even broader phenotypic spectrum, including early developmental delay, epileptic encephalopathy. Those mutations' relationship to disease are not easily explained by simple change to the alpha-1A that alters calcium channel properties. Those uncertainties bring us to recognize a de novo CACNA1 gene encoded protein alpha-1A. Around 2006, when we studied the pathogenesis of SCA6, we recognized that there is a 17.5 kilodalton C-terminal fragment of alpha-1A subunit present physiologically as a stable protein in the cerebellar Purkinje cell. We named it alpha-1ACT. Later, we discovered that mechanism controlling alpha-1ACT expression by showing that CACNA1 mRNA contains a secondary RNA structure that is internal ribosome entry site arrays. Ribosome recognize and bound to arrays initial cap-independent translation, which is a non-classical translation, as a part of greater process of protein synthesis is well known in virus. Alpha-1ACT is one of a few proteins that are generated by the cellular arrays and is a second protein product of CACNA1 gene. Interestingly, the polyQ expansion is presence within two independently translated but functionally unrelated gene products, alpha-1A and alpha-1ACT. Whether alpha-1A plays a role in pathogenesis of SCA6? After we reported, SCA6 researchers in Australia and Japan also identified that polyQ expanded alpha-1ACT produces cell toxicity and exhibits reduced transcription factor function in SCA6 patient-derived iPSC or differentiated Purkinje cells. our lab established a alpha-1 ACT SCA6 mouse model, targeting expression alpha-1 ACT SCA6 in cerebellum of Purkinje cell. We found that alpha-1 ACT SCA6 mice had progressive motor impaired and Purkinje cell atrophy, as shown by the immunofluorescence on the top and the electron microscopy at the bottom. Under the normal condition, you can see the healthy structure of Purkinje cell, but it dissolved in SCA6. This result suggests that alpha-1 ACT may drive the pathogenesis of SCA6. Now we move from the background to our main topic, alpha-1 ACT and development. The numerous clinical studies have reported that supply-side mutation, deletions, and frame shift distributed in the CAGNA1 areas or alpha-1 ACT coding region were correlated with developmental disorder. With these clinical findings, we want to explore what is the physiological function of native alpha-1 ACT in early life, and whether it is a possible relationship between neurodevelopmental disorders and regulation of alpha-1 ACT. To study the function of alpha-1 ACT, we established a Keiko mouse model. We crossed alpha-1A knockout mice with humanized CAGNA1 exon 47 mice. The final mice model have one humanized CAGNA1 gene allele and one non-allele. We name it Keiko. The Keiko mice have a 90% selective decreased alpha-1 ACT expression at both mRNA and protein level. The survival rate of Keiko mice is around 20% with the routine animal care. The body weight of survived Keiko mice decreased 60%. As we show in this video, we recorded from the Keiko mice at P18 and 1 month old. The mice in the middle is the Keiko mice. The Keiko mice have, we can observe seizure, dystonia, and ataxia from those mice. This is P13. This one is Keiko. The impaired mobility were further validated in the rotorage and the open field test. To test whether the absence of alpha-1 ACT is a critical factor responsible for poor survival and delayed motor development in Keiko mice, we crossed our transgenic mice into Keiko mice and generate a mice lines that selectively expresses only alpha-1 ACT in Purkinje cells of Keiko mice. We name it Keiko alpha-1 ACT. From previous video, we know the one have seizure is Keiko. The third mice we put in is Keiko rescue. The survival rate were fully recovered and the body weight were significantly increased by selective expression alpha-1 ACT. As shown in this video, motor abnormalities in Keiko mice were improved in Keiko alpha-1 ACT. This suggests that alpha-1 ACT targeted to Purkinje cell plays an important role in neonatal survival and cerebellar development. After this observation, we call Keiko alpha-1 ACT as Keiko rescue. Because the transgenetic expression alpha-1 ACT in Purkinje cell under the TETOF system, we were able to use doxycycline to inhibit alpha-1 ACT expression in Keiko rescue mice. To further investigate temporal effect of alpha-1 ACT expression, we use doxycycline to inhibit expression of alpha-1 ACT transgene in Keiko rescue mice at different life stage. The effect of inhibition were tested over 4 periods: prenatal, early, middle, and late. Interestingly, we found that alpha-1 ACT mediated Keiko rescue is time-dependent. With the prenatal inhibition of alpha-1 ACT expression, alpha-1 ACT mediated the restored phenotype was not observed. The survival rate, body weight, and local motor function of Keiko rescue mice were the same as those of Keiko. Moreover, even after doxy treatment was discontinued after 1 month of age, allowing alpha-1 ACT expression to be re-established, phenotypes of Keiko rescue mice aged 1 to 12 months were still very similar to age-matched Keiko mice. Based on this data, it suggests that loss of alpha-1 ACT in prenatal period induced the irreversible developmental disability. When alpha-1 ACT inhibition was initiated at 1 month or 3 months old, the motor abilities of Keiko rescue mice were impaired compared with those no inhibition intermediate, but not as severe as those observed in prenatal inhibition. Surprisingly, if alpha-1 ACT inhibition was initial at 6 months, there were no significant difference in motor behaviors between Keiko rescue mice with and without exposed to doxy. Keiko rescue mice with inhibited alpha-1 ACT expression initial at 6 months do not suppress restored motor activity. Thus, the presence of alpha-1 ACT specifically within the prenatal period is pivotal for early cerebellar development. Inhibition of alpha-1 ACT expression within the 1st to 3rd months of life, but not at the later stage of life, has significantly impacted at motor function. This result indicates that alpha-1 ACT operates in a highly age-dependent temporal window. We previously showed that alpha-1 ACT is a transcription factor that regulates the expression of several neuronally expression gene, promotes the neurite outgrowth. To explore the physiological gene regulation properties of alpha-1 ACT, we performed ChIP-seq and RNA-seq study. ChIP-seq identified that alpha-1 ACT occupied genomic region. This figure shows that alpha-1 ACT binding site distribution is around 3,000 base pair related to the transcription start site. To monitor transcriptional profiles that capture transient and prolonged dynamic change regulated by alpha-1 ACT, we performed a time series RNA-seq study. The heat map shows the direct indirect regulation target gene identified by RNA sequence. Alpha-1 ACT integrated ChIP-seq and RNA-seq differentially expression genes were also identified as shown in bottom figure. To identify how the whole transcriptional network correlated with the biological function of alpha-1 ACT, we applied co-expression network and GO-term analysis with the R1272 RNA sequence identified gene. This reveals that alpha-1 ACT target DEGs are significantly enriched in the neurogenesis and synaptic signaling. 39 alpha-1 ACT specific persistent DEG were identified across the 4 time points. GO-term analysis for 39 gene reveals that top 10 enriched GO-term also were related to the neuronal developments and synaptic signaling. The circle indicates the distribution of 39 genes in different GO-term. 21 of 39 DEGs were ChIP-seq targets, which are in the center yellow circle. To verify that alpha-1 ACT specific neurogenesis gene identified in cell were expressed in cerebellar Purkinje cells, we performed laser capture, microdissection, and extract the RNA from Purkinje cell with control Keiko and Keiko rescue mice. Expression alpha-1 ACT neurodevelopment related target gene is decreased in Keiko mice, but restored in Keiko rescue mice. We also explored the temporal transcriptal profiles of alpha-1 ACT in human cerebellum at different ages. In human, CACNA1 mRNA expression were maximal from birth to age of 20 years and decreased gradually until reach a plateau at age 50. The expression of alpha-1 ACT target genes in human cerebellum can be grouped into 4 major dynamic expression patterns. Those involved in neurodevelopment were highly expressed at 1 month and start to decrease with aging after maturation. Those associated with synaptic targeting were highly expressed at term and decreased with age gradually. Combine the temporal change of alpha-1 ACT transcriptome with an increasing number of clinical reports of pediatric patients with developmental delay and cerebellar atrophy that were associated with large deletion or mutation involved in the C-terminal of CACNA1 gene. It reveals that impaired alpha-1 ACT expression or function may disturb the gene expression network within cerebellum. In conclusion, we have shown that the gene CACNA1A encodes alpha-1 A subunit of neuronal calcium channel PQ. Due to presence of a novel arrays within the arrays, the mRNA, CACNA1 also encodes a second unrelated protein, alpha-1 ACT, with distinct function as a transcription factor. We also discovered that alpha-1 ACT is essential for survival and cerebellar programming in early life by orchestrating a network driving neurogenesis to mediate maturation of Purkinje cell and the cerebellum. Each of neurological disease is associated with a different CACNA1 mutation, suggesting a differential effect on calcium signaling and alpha-1 ACT transcription factor function. Our finding proposes that bicistronic gene may allow differential but coordinated control of gene expression for two functionally unrelated gene products, and there exists may help explain the complex genetic syndromes associated with mutations in those genes. So I would like to thank Dr. Gomez and everyone in our lab. Without their hard work and dedication, we won't be able to continue our journey with alpha-1 ACT. I also thank you for your attention. I would like to take any question you have.